A simple guide to CRISPR, one of the biggest science stories of the decade. It could revolutionize everything from medicine to agriculture. Better read up now. Now, keep in mind, this is from Vox, so yes, it's true what's in this article, but it is like kind of a goofy little article. This was updated at the end of 2018, so relatively recently. One of the biggest and most important science stories of the past few years will probably also be one of the biggest science stories of the next few years. So this is as good a time as any to get acquainted with the powerful new gene editing technology known as CRISPR. If you haven't heard of CRISPR yet, the short explanation goes like this. In the past 9 years, scientists have figured out how to exploit a quirk in the immune systems of bacteria to edit genes in other organisms. Plants, mice, even humans. With CRISPR, they can now make these edits quickly and cheaply, in days rather than weeks or months. The technology is often known as CRISPR-Cas9, but we'll stick with CRISPR. Pronounced CRISPR. That's helpful if you're reading it, but I already knew it was pronounced CRISPR. We're talking about a powerful new tool to control which genes get expressed in plants, animals, and even humans. The ability to delete undesirable traits and potentially add desirable traits with more precision than ever before. So far, scientists have used it to reduce the severity of genetic deafness in mice, suggesting it could one day be used to treat the same type of hearing loss in people. They've created mushrooms that don't brown easily, and edited bone marrow cells in mice to treat sickle cell anemia. Down the road, CRISPR might help us develop drought-tolerant crops and create powerful new antibiotics. CRISPR could one day even allow us to wipe out entire populations of malaria-spreading mosquitoes, or resurrect once extinct species, like the passenger pigeon. Of all the things to resurrect, passenger pigeons. A big concern is that while CRISPR is relatively simple and powerful, it isn't perfect. Scientists have recently learned that the approach to gene editing can inadvertently wipe out and rearrange large swaths of DNA in ways that may imperil human health. That follows recent studies showing that CRISPR-edited cells can inadvertently trigger cancer. That's why many scientists argue that experiments in humans are premature. The risks and uncertainties around CRISPR modification are extremely high. On this front, 2018 brought some shocking news. In November, a scientist in China, I don't know how to pronounce it, but He Jiankui, reported that he had created the world's first human babies with CRISPR-edited genes, a pair of twin girls resistant to HIV. The announcement stunned scientists around the world. The director of the National Institutes of Health, Francis Collins, said the experiment was profoundly disturbing and tramples on ethical norms. It also created more questions than it answered. Did Jiang Kui actually pull it off? Does he deserve praise or condemnation? Do we need to pump the brakes on CRISPR research? There's a video here that I would suggest watching if you'd like to. I'm not going to play it right now, but it is a pretty decent video. While independent researchers have not yet confirmed that Jiankui was successful, there are other CRISPR applications that are close to fruition, from new disease therapies to novel tactics for fighting malaria. So here's a basic guide to what CRISPR is and what it can do. Another video worth watching if you would like to see a visual of how this genome editing works. If we want to understand CRISPR, we should go back to 1987, when Japanese scientists studying E. coli bacteria first came across some unusual repeating sequences in the organism's DNA. The biological significance of these sequences, they wrote, is unknown. Over time, other researchers found similar clusters in the DNA of other bacteria and archaea, another type of bacteria. They gave these sequences a name: clustered, regularly interspaced, short palindromic repeats, or CRISPR. Yet the function of these CRISPR sequences was mostly a mystery until 2007, when food scientists studying the Streptococcus bacteria used to make yogurt showed that these odd clusters actually served a vital function. They're part of the bacteria's immune system. See, bacteria are under constant assault from viruses, so they produce enzymes to fight off viral infections. Whenever a bacterium's enzymes manage to kill off an invading virus, other little enzymes will come along, scoop up the remains of the virus's genetic code, and cut it into tiny bits. The enzymes then store those fragments in CRISPR spaces in the bacterium's own genome. Now comes the clever part. CRISPR spaces act as a rogues gallery for viruses, and bacteria use the genetic information stored in these spaces to fend off future attacks. When a new viral infection occurs, the bacteria produce special attack enzymes, known as Cas9, that carry around those stored bits of viral genetic code like a mugshot. When these Cas9 enzymes come across a virus, they see if the virus's RNA matches what's in the mugshot. If there's a match, the Cas9 enzyme starts chopping up the virus's DNA to neutralize the threat. It looks a little like this. That's a picture, if you weren't following along with the video. So that's what CRISPR-Cas9 does. For a while, these discoveries weren't of much interest to anyone except microbiologists, until a series of further breakthroughs occurred. How did CRISPR revolutionize gene editing? In 2011, Jennifer Doudna of the University of California, Berkeley, and Emmanuel Charpentier of Umeå University in Sweden were puzzling over how the CRISPR-Cas9 system actually worked. How did the Cas9 enzyme match the RNA in the mugshots with that in the viruses? How did the enzymes know when to start chopping? The scientists soon discovered they could fool the Cas9 protein by feeding it artificial RNA, a fake mugshot. When they did that, the enzyme would search for anything with that same code, not just viruses, and start chopping. In a landmark 2012 paper, Doudna, Charpentier, and Martin Janek showed they could use this CRISPR-Cas9 system to cut up any genome at any place they wanted. While the technique had only been demonstrated on molecules in test tubes at that point, the implications were breathtaking. Further advances followed. Feng Zhang, a scientist at the Broad Institute in Boston, co-authored a paper in Science, which is a really big science paper, like where you could get published. That's like huge. In February 2013, showing that CRISPR-Cas9 could be used to edit the genomes of cultured mouse cells or human cells. In the same issue of Science, Harvard's George Church and his team showed how a different CRISPR technique could be used to edit human cells. Since then, researchers have found that CRISPR-Cas9 is amazingly versatile. Not only can scientists use CRISPR to silence genes by snipping them out, they can also harness repair enzymes to substitute desired genes into the hole left by the snippers, though this latter technique is trickier to pull off. So for instance, scientists could tell the Cas9 enzyme to snip out a gene that causes Huntington's disease and insert a good gene to replace it. Gene editing itself isn't new. Various techniques to knock out genes have been around for years. What makes CRISPR so revolutionary is that it's so precise. The Cas9 enzyme mostly goes wherever you tell it to, and it's incredibly cheap and easy. In the past, it might have cost thousands of dollars and weeks or months of fiddling to alter a gene. Now, it might cost just $75 and only take a few hours. And this technique has worked on every organism it's been tried on. It's now one of the hottest fields around. In 2011, there were fewer than 100 published papers on CRISPR. In 2018, there were more than 17,000 and counting. With refinements to CRISPR, new techniques for manipulating genes, improvements in precision, and more, this has become such a fast-moving field that I even have trouble keeping up now, says Doudna. We're getting to the point where the efficiencies of gene editing are at levels that are clearly going to be useful therapeutically, as well as a vast number of other applications. There's also been an intense legal battle over who exactly should get credit for this CRISPR technology, and who owns the potential lucrative rights. Was Joanna's 2012 paper at the University of California, Berkeley the breakthrough, or was Zhang's 2013 research at the Broad Institute the key advance? In September, a federal appeals court rejected the University of California, Berkeley's arguments that the school has exclusive rights to CRISPR patents, upholding the Broad Institute's patents on some CRISPR applications. But the important thing? CRISPR has arrived. So what can we use CRISPR for? So many things! Paul Knopfler, an associate professor at UC Davis School of Medicine, told Vox that CRISPR makes him feel like a kid in a candy store. At the most basic level, CRISPR can make it much easier for researchers to figure out what different genes in different organisms actually do. By, for instance, knocking out individual genes and seeing which traits are affected. This is important. While we've had a complete map of the human genome since 2003, we don't really know what function all those genes serve. CRISPR can help speed up genome screening, and genetics research can advance massively as a result. Researchers have also discovered that there are numerous CRISPRs. So CRISPR is actually a pretty broad term. It's like the term fruit. It describes a whole category. Said the Broad's Zhang. When people talk about CRISPR, they're usually referring to CRISPR-Cas9 system we've been talking about here. But in recent years, researchers like Zhang have found other types of CRISPR proteins that also work as gene editors. Cas13, for example, can edit DNA's sister, RNA. Cas9 and Cas13 are like apples and bananas, Zhang added. The real fun, and potentially the real risks, could come from using CRISPRs to edit various plants and animals. A 2016 paper in Nature Biotechnology by Rodolphe Berengo and Duadna listed a flurry of potential future applications. Number one is edit crops to be more nutritious. Crop scientists are already looking to use CRISPR to edit the genes of various crops, to make them tastier, or more nutritious, or better survivors of heat and stress. They could potentially use CRISPR to snip out the allergens in peanuts. Korean researchers are looking to see if CRISPR could help bananas survive a deadly fungal disease. Some scientists have shown that CRISPR can create hornless dairy cows, a huge advance for animal welfare. Recently, major companies like Monsanto and DuPont have begun licensing CRISPR technology, hoping to develop valuable new crop varieties. While this technique won't entirely replace traditional GMO techniques, which can transplant genes from one organism to another, CRISPR is a versatile new tool that can help identify genes associated with desired crop traits much more quickly. It could also allow scientists to insert desired traits into crops more precisely than traditional breeding, which is a much messier way of swapping in genes. With genome editing, we can absolutely do things we couldn't do before, says Pamela Ronald, a plant geneticist at the University of California, Davis. That said, she cautions that it's only one of many tools for crop modification out there, and successfully breeding new varieties could still take years of testing. It's also possible that these new tools could attract controversy. Foods that have had genes knocked out via CRISPR are currently regulated more lightly than traditional GMOs. Policymakers in Washington, D.C. are currently debating whether it might make sense to rethink regulations here. This piece for Enzia by Mewa Montenegro delves into some of the debates CRISPR raises in agriculture. Number 2, new tools to stop genetic diseases. Scientists are now using CRISPR-Cas9 to edit the human genome and try to knock out genetic diseases like hypertrophic cardiomyopathy. They're also looking at using it on mutations that cause Huntington's disease or cystic fibrosis, and are talking about trying it on the BRCA1 and BRCA2 mutations linked to breast and ovarian cancers. Scientists have even shown that CRISPR can knock HIV infections out of T cells. So far, however, scientists have only tested this on cells in the lab. there are still a few hurdles to overcome before anyone starts clinical trials on actual humans. For example, the Cas9 enzymes can occasionally misfire and edit DNA in unexpected places, which in human cells might lead to cancer or even create new diseases. As geneticist Alan Bradley of England's Wellcome Sanger Institute told Stat, CRISPR's ability to wreak havoc on DNA has been seriously underestimated. And while there have also been major advances in improving CRISPR precision and reducing these off-target effects, scientists are urging caution on human testing. That's a big reason why Jiankhu's experiments in producing human babies with CRISPR-edited genomes are so controversial and alarming. Researchers who examined the few findings that Jiankhu publicly revealed said that the results show that the baby's genes were not edited precisely. There's also plenty of work to be done on actually delivering the editing molecules to particular cells, a major challenge going forward. Number 3, powerful new antibiotics and antivirals. One of the most frightening public health facts around is that we are running low on effective antibiotics, as bacteria evolve resistance to them. Currently, it's difficult and costly to develop fresh antibiotics for deadly infections, but CRISPR-Cas9 systems could, in theory, be developed to eradicate certain bacteria more precisely than ever. Though again, figuring out delivery mechanisms will be a challenge. Other researchers are working on CRISPR systems that target viruses, such as HIV and herpes. And number 4, gene drives that could alter entire species. Scientists have also demonstrated that CRISPR could be used, in theory, to modify not just a single organism, but an entire species. It's an unnerving concept called gene drive. It works like this. Normally, whenever an organism, like a fruit fly, mates, there's a 50-50 chance that it will pass on any given gene to its offspring. But using CRISPR, scientists can alter these odds so that there's nearly a 100% chance that a particular gene gets passed on. Using this gene drive, scientists could ensure that an altered gene propagates throughout an entire population in short order. By harnessing this technique, scientists could, say, genetically modify mosquitoes to only produce male offspring, and then use a gene drive to push that trait through an entire population. Over time, the population would go extinct. Or you could just add a gene making them resistant to the malaria parasite, preventing its transmission to humans, Vox's Dylan Matthews explains in his story on CRISPR gene drives for malaria. Suffice to say, there are also hurdles to overcome before this technology is rolled out en masse, and not necessarily the ones you'd expect. The problem of malaria gene drives is rapidly becoming a problem of politics and governance more than it is a problem of biology, Matthews writes. Regulators will need to figure out how to handle this technology, and ethicists will need to grapple with knotty questions about its fairness. Number 5. Creating designer babies. This is the one that gets the most attention, and rightly so. It's not entirely far-fetched to think we might one day be confident enough in CRISPR's safe to use it to edit the human genome, to eliminate disease, or to select for athleticism or superior intelligence. But Jiankhu's recent attempt to introduce protection from HIV in embryos intended for pregnancy, which involved little oversight, is not how scientists want the field to move forward. The concern is that there are not enough safeguards yet to prevent harm, nor enough knowledge to do definite good. In Jiankhu's case, he also did not tell his university about his experiment ahead of time, likely did not inform the parents of the modified babies of the risks involved, and may have had a financial incentive from his 2 affiliated biotech companies. We're not even close to the point where scientists could safely make the complex changes needed to, for instance, improve intelligence, in part because it involves so many genes. So don't go dreaming of Gattaca just yet. I think the reality is we don't understand enough yet about the human genome, how genes interact, which genes give rise to certain traits, in most cases to enable editing for enhancement today, Duana said in 2015. Still, she added, that'll change over time. Wait, should we really create designer babies? Given all the fraught issues associated with gene editing, many scientists are advocating a slow approach here. They are also trying to keep the conversation about this technology open and transparent, build public trust, and avoid some of the mistakes that were made with GMOs. But with CRISPR's ease of use and low cost, it's challenging to keep rogue experiments in check. In February 2017, a report from the National Academy of Sciences said that clinical trials could be greenlit in the future for serious conditions under stringent oversight. But it also made clear that genome editing for enhancement should not be allowed at this time. Society still needs to grapple with all the ethical considerations at play here. For example, if we edited a germline, future generations wouldn't be able to opt out. Genetic changes might be difficult to undo. Even this stance has worried some researchers like Francis Collins of the National Institutes of Health, who has said that the U.S. government will not fund any genomic editing of human embryos. In the meantime, researchers in the U.S. who can drum up their own funding, along with others in the UK, Sweden, and China, are moving forward with their own experiments.